3, 2. Hello, everyone. Thank you very much for joining us on the first day of this Physio webinar spin-off in advanced topics in thermal biology. This event is organized by graduate students from the graduate program in general physiology at the Institute of Biosciences of the University of São Paulo. Today, we will attend 6 talks over 2 days, 3 today, with the participation of renowned speakers from different countries talking about different aspects of thermal biology. This event is being live streamed on our YouTube channel and will be recorded. And you can watch lectures from our past Physio webinars editions on our YouTube channel too. Please follow us. Today, we will have, as I said, 3 talks. And after each talk, everyone in the audience on YouTube can ask questions through the chat. You can also send your questions in Portuguese if you like, and our team will select them and so I can read to the speakers in English. Okay, so without any more preamble, we are very pleased to welcome our first speaker, Dr. Volker Loeschcke. He holds a PhD in genetics from the Free University of Berlin, and he's currently a full professor at the Department of Biology, section of Genetics, Ecology, and Evolution at Aarhus University. His research is focused on adaptations to stressful environments, combining approaches from evolutionary, genetics, ecophysiology, and molecular biology using Drosophila as a model. So, Dr. Volker Loeschcke, thank you very much for your presence here today. After your talk, we will have up to 10 minutes to take questions from the audience. Thank you very much.
Okay, thank you, Gustavo, for the nice introduction. I have to apologize that my voice is not very good. So, as I had a cold the last 8 to 10 weeks, and this somehow affected my voice. But let's hope that you understand me anyway. I want to talk about some work that has been done in my lab for the last 15 years with many colleagues, former PhD students, postdocs, and others, and which was supported by many funding organizations as the National Science Research Council and other foundations. I want to talk about costs and benefits of plastic versus evolutionary responses to environmental stress. We can ask, what is stress in a way? And stress can be competition, predation, drought, high mental work, genetic stress as inbreeding, pesticides, alcohol, you name it. But for evolutionary studies, a useful definition of environmental stress is any environmental change that drastically reduces fitness of an organism and stimulates physiological, metabolic, or behavioral adjustments in body function, a definition by Hoffman and Parsons from 1997. If we look at temperature as one stressor, we can see that fitness is dramatically reduced in the orange ranges of the temperature scale. And this reduction is different for the fitness components, fertility and viability. If we look at some real data from Drosophila melanogaster, we can see that temperatures above, and that is developmental temperatures, above 14 or so and 30 do not much influence viability. But if you get below 14 and above 32, you get stressful conditions and you get dramatically reduced viability. Under natural conditions, temperatures are not constant, and we have always a kind of time component with stress levels determined by temperature times exposure time. If we now use this setup and think about possible responses to reduce the detrimental impact of thermal stress or of other stress factors, we differentiate between plastic responses induced by acclimation or hardening or just by the environmental conditions, as a response within generations, as a here and now response, and evolutionary adaptation as a kind of longer term across generation response. We of course have also behavioral avoidance. You just move away from the stress if you can. And this behavioral avoidance can also have an evolutionary part and can evolve, as well as the plasticity can evolve. We use Drosophila sorry, as a model organism, not only because it has minimal space requirements, a short generation time, and is easy to handle and cheap, but because Drosophila has a long tradition for genetic studies. Its genome has been mapped already in the year 2000. And what also is interesting in Drosophila, that you have many different species, cosmopolitan as well as specialist species. And so Drosophila also shows a large similarity between flies and vertebrates on the cellular level. Let us take an example for the critical thermal minimum, CT-min, as a function of developmental acclimation temperature, which means of the temperature flies developed from egg to adults. If we look at what happens with CT-min, when in temperatures from 13 to 30 centigrades, we see that the critical thermal minimum changes by 6 centigrades. So we have an enormous plasticity for this trait. If we instead look at the critical thermal maximum, CT max, for flies developmentally acclimated at different temperatures, we find that for the same temperature range from 13 to 30, that the difference in plasticity is much less for the critical thermal maximum than for the critical thermal minimum. So we have less plasticity in this thermal maximum. A mechanism behind that has been described in the 80s, but is more and more coming, is a mechanism behind the heat hardening response is a heat shock response, which is the induction of heat shock proteins, which we also call HSPs, that protect cells from damage by refolding denaturated proteins, but also by working as a kind of housekeeping system by turning damaged proteins to the intercellular degradation system. If we look in this under this background at costs of this plasticity, we can look at the number of offspring produced per day during days 1 to 2, 3 to 4, 5 to 6, and so on. And we can see that in the first 2 days, the difference in offspring produced of flies that were hardened by a 1 hour 37-degree treatment, as compared to the red line, the no hardened flies, we got a 50% decrease in fecundity by the hardening treatment. So we got quite some strong hardening costs. If we look a little broader on acclimation treatment and how it affects resistant traits, we look at this figure and say that if we acclimate by cold, then cold resistance is positively affected. If we acclimate by heat, then heat shock resistance is positively expected. So in the diagonal, we acclimate by the same treatment trait as we look at resistance afterwards, and we get also only pluses. But if we compare outside of the diagonal, we see that we find many more minus signs in this figure than plus signs, meaning that if we, for example, acclimate by cold, heat shock resistance is reduced significantly, and starvation resistance also is reduced significantly. Similarly, if we acclimate for heat, we can see that desiccation is reduced, desiccation resistance is reduced significantly, and starvation resistance is reduced significantly. So we get clear reductions we get a clear cost of acclimation treatment, particularly if we acclimate for others than for the traits that we test. If we want to see how this data fits to natural conditions, how costly the acclimation is under semi-natural conditions, we expose flies, we release flies at a release point and recapture them afterwards from buckets that are 5, 10, 15 meters and so on apart. And we use fitness as a kind of ability to re-find resources, which are buckets with mashed bananas. And if we do that, for example, if we acclimate at 15 centigrades during development, this open square, we find that at temperatures of around 12 to 13 centigrades, the acclimated flies perform 100 times better than the control flies, which are normalized to this one line. So we get an enormous benefit of this acclimation treatment of cold acclimated flies at cold temperatures, while the same acclimated flies at warm temperatures show a 30 times decrease in performance. Similarly, if we, for example, take the 36 degrees adult acclimation, the hot acclimation as the black squares, we can see that at very hot temperatures, these flies perform 30 times as good as the controls. And at the cold temperatures, we don't have data here, but it seems that they go also into the range of negative values. So in a way, we can say that plasticity also under natural or semi-natural conditions is costly. So if we sum up, the effect of hardening and acclimation can have large beneficial effects if hardening or acclimation and stress type are similar, but otherwise they may have strong negative side effects, or you could say costs, with respect to traits other than those acclimated for. So thermal adaptation based on plasticity can be very costly. If we now look at the other side of the medal, namely at evolutionary adaptation, by looking at selection lines selected to improve tolerance to environmental stress, as alternative to plastic responses, possibly what we want to look at lower costs. We have lines, three heat selection regimes: heat resistance in the water bath, survival at constant 30 degrees, or heat knockdown resistance in knockdown tubes, where we always look at 5 biological replicates with larger than 300 pairs per replicate, and we select also for cold resistance, starvation resistance, desiccation resistance, and longevity, as longevity earlier has been shown to be related to some of the resistance traits. If we look at the results from this selection experiments, we find that CS cold selected lines are more cold shock resistant than controls, which are normalized at the 0 line, but also more heat selected, HS heat selected, knockdown selected, or desiccation selected flies are more cold shock resistant than controls. So we get a kind of cross selection benefit, or a kind of general stress response. You can do the same for the heat selected HS lines and while looking at heat shock resistance. And again, we find that KS lines, knockdown selected, and DS desiccation selected lines also perform significantly better than the control lines. We have a similar pattern also for desiccation resistance, DS, where the desiccation selected lines perform better under desiccation conditions. And for starvation resistance, we see that starvation selected SS lines perform best, but also a couple of other selection lines are more starvation resistant than control lines. The two subfigures to the right, heat knockdown resistance and longevity selection, does not show such a general response. For longevity selection, we can say here we have only 7 generations of selection, while for the other traits, we have 20 generations of selection. And for heat knockdown resistance, this apparently is related to a muscle that somehow appears to stop functioning at around 30 centigrades. So in a way, we can say we get a kind of lots of cross benefits of selection. If we look at developmental time, we can say that flies that were selected for the different traits on average develop slower, use more time to develop from egg to adult. And if we look at thorax length as a size component, we see that most lines are slightly smaller, but this is not significant with exception for the longevity selected lines. So in a way, we have also some costs, but not very strong costs of selection for the different environmental the traits. And if we now try to summarize, we can say that selection for stress-resistant traits usually leads to positive correlated responses in other stress-resistant traits without any obvious larger costs. So we have a kind of general stress response on the phenotypic level. And even so I haven't shown much of it, we can say that this selection for stress resistance leaves strong signatures on gene expression, metabolic profile, and the proteome. And indicates strongly the existence of a general stress response based on data from the proteome, transcriptome, and metabolome. And finally, demonstrates an important role of the heat shock proteins for the general stress response. So I thank you for your attention, and I thank my colleagues that are named here and many others that have contributed to this work for their contribution. And I also thank the different funding organizations for support, financial support to these studies. I think my time is more or less gone, so I thank you for your attention.
Well, thank you very much, Dr. Volker Loeschcke, for your very interesting presentation. Now we will take some questions from the audience. Please, all of you who are in the audience, please send your questions through the chat on YouTube. Send them in Portuguese if you like, and our staff will send them and we can read them in English for Dr. Volker. So there's a first question here. How does heat knockdown work at the molecular level, and if there is any difference in number of offspring compared to the control group in your acclimation experiments?
I'm not quite sure. The heat knockdown flies are selected in what we call a knockdown tube, where we have a warm temperature room or a water bath that warms up flies in the tube and where they fall down. So they, so the offspring of these flies are probably not much affected. But I didn't get the other part of the question. Could you?
The other
repeat?
part, yeah, if you know about the underlying molecular mechanisms when this heat knockdown response occurs, like what happens at the molecular level in the flies?
Basically, that is what we try to relate the phenotypic traits with the molecular background. But in this case, there is not much of a strong response on the level of the heat shock proteins. We have done, and I could go back to the screen, we have done sequencing and have sequenced all the lines and have found a lot of candidate genes for the different traits. But I thought that would have been too detailed to tell here. Let me just share screen and see if.
Yeah, please go ahead.
Share. Oops. Why does it? Okay. Here we have these different lines, which we then, where we get SNPs and get about 100,000 SNPs, comparing the founder population with the unselected controls and with the selection lines. And we get a strong result. We get lots of genes that are shared by the different replicate lines. And that means also we get this for the heat knockdown selected lines. So we get distinct genomic fingerprints from all selection regimes. Yes.
Great, thank you. We have another question here. Someone would like to know if this program that you showed in Drosophila as a model to study thermal biology, what are the considerations to apply this to other insect species, or what we could learn from Drosophila, from the specificities of Drosophila to other insect species?
I think when we use the model organism, we hope that the results we get have some meaning for other organisms. Drosophila are much easier to handle, and there's much more experience. But I think it's a good idea to try to do it, to perform similar experiments on other ectothermic organisms. And many of the results should be similar. And results include that chronic stress is always bad, and I guess that similar genes in different organisms will be upregulated under particular stress types. So there you could do a comparison between the gene responses, the genomic responses following stress exposure. And that is what should be followed up after you have done these things. And that is what we have also in these other studies that I shortly mentioned, followed up. Yeah.
Thank you.
Sorry.
Well, I have a question on my own. You showed that the acclimation response is higher when we compared cold experimental regimes versus heat, right? That the acclimation response is higher. So what could we infer from these results you showed on that regard for scenarios of climate warming? If you understood me.
I think the difference between being alive and dead is much shorter on the hot side than on the cold side. So you have a much longer tail of resistance or of tolerance at the cold side than you have it on the heat side. So you cannot increase temperatures too much before you get very drastic effects. So in that sense, that's probably not good on the hot side that you have climate change. Yes.
Okay, thank you. There is another question here. It says the traits in your selection lines, the tests that you selected under were stress tolerance traits, the traits you compared in your selection lines, and you find little cost across traits. Do you think you would have found cost if you had used fertility traits instead of a stress tolerance trait under heat or cold?
Yes, we have shown under heat that you get effects on fertility. And I think that's part of the game, that you look at the different types of traits, and fertility traits. I think I showed a slide were affected by hot temperatures, and I think they will also be affected by other stress traits. But in a way, you could say the other cross-trait responses were usually positive, but fertility is a different type of trait. So yes, I think I would expect that some of the fertility or reproductive traits would be negatively affected by exposure to environmental stress.
Yeah, thank you. Well, there's another question regarding, you showed that the recapture rate for flies that were acclimated to thermal stress was higher than in control lines, but it was effects of acclimation, right? Have you performed some experiments like that on recapturing flies, but that were under selection regimes?
We have oops. Can you see something?
Not yet.
Okay. then I don't have properly shared the screen. Can you see the slide now?
Not yet, Volker. If you want to share the screen.
I thought
yeah.
I thought I had shared the screen. Okay, and it now doesn't function. I can say yes. We released heat selected, cold selected, and other selected flies out in the wild and recaptured them, and we could see that heat selected flies were found recaptured much more often at hot temperatures, while cold selected lines were more often recaptured under cold conditions. So in that sense, yes, we got also responses of the selection lines under natural conditions.
Okay, that's great. I don't think we have more questions, so I thank you again, Dr. Volker. Now we will have a short break of 5 minutes. Well, actually, we will start our next talk at 8:40 in Brazil, in São Paulo time. And we continue with the next speaker. So thank you very much, Dr. Volker Loeschcke, for your participation.
It was a pleasure.
Hello again. Now we are back and ready for our second speaker. We welcome Dr. Jesper Givskov Sørensen. He holds a PhD degree from the Department of Biological Sciences at Aarhus University in Denmark, where he is a full professor also now. His research is about adaptation to environmental stressful condition in terrestrial invertebrates, with special attention to effects of high and low temperatures. So Jesper, thank you very much for your presence here today. After your talk, we will have up to 10 minutes again to take questions from the audience. And yeah, thank you.
Thank you, Gustavo. Let me share my screen. All right. So can you confirm that everything looks okay, Gustavo?
Yes, it does.
All right. Thank you very much for this opportunity. It's a really nice opportunity to test out this format as well. And thanks to you guys for organizing this. For those who saw the last presentation by Volker, then I should say that I'm a colleague of Volker and have worked together with him for quite a long time and have shared a lot of the same interests. So it's not my intention to argue that behavior for thermal biology is more important than physiology or evolution, but that we need to consider behavior as well. So I made this slightly provocative title saying that, or asking what little do we actually know about the role of behavior for thermal adaptation and responses in small ectotherms. So first of all, what do we know about behavior? In larger ectotherms, mainly lizards, the behavior in terms of temperature biology have been studied a lot, and we know a lot about how the behavior plays a huge role for these organisms to attain a good temperature where the body functions well. And behavioral responses are, of course, really fast. The first thing a small insect will do if it's find itself in a too warm or too cold environment is that it might try to move. So these behavioral responses can act really fast. We also know that thermal heterogeneity can be considerable on short temporal and spatial scales. And if you have an adaptation or an adaptive response in your physiology that does not match the environment you're in, it can be maladaptive. So there's a risk that this heterogeneity in the environment makes any responses that are slightly longer term maladaptive. And their behavior again could play a role. Even if we know this, then behavioral aspects of thermal biology of insects is not very well investigated and understood compared to the ecophysiology of these animals. And I will show you 3 examples of studies where behavior might be important for our understanding, where behavioral responses might interact with evolutionary adaptation or with plastic physiological responses in complex ways. And argue through those that we need to think about behavior in some cases at least, and consider it more when we work with thermal biology. So first of all, one could ask what are small ectotherms actually adapted to or exposed to in the field. And for a long time and for good reasons, we have considered the environment of the world as represented here to the left. So the black dots are annual mean temperatures measured across many different latitudes. That comes from a study by Chown et al. In 2004. And we know, of course, that around 0 latitudes, we have higher temperatures, and the temperatures on average decrease towards, in this case, the left here is the northern hemisphere and the right is the southern hemisphere. And we can, of course, see that yes, in Denmark, it's colder around here than it is in the tropics on average. The other dots, the open symbols, they symbolize the warmest and the coldest temperatures measured in this 1 year. But and there's also some variation, but it's behaving slightly different than the annual mean, so that the maximum temperatures measured are more uniform around the world. But this variation is not what's relevant for small insects. And a number of studies, including the Potter study mentioned here, have argued that that microclimatic variation actually might exceed the geographic variation that we are working with in a lot of the macroecological studies that are being published. So this is just to the right here, a picture that I took yesterday out the window of the university building. And we see some bushes, some trees, we see some open grass, and there's sun and there's shade, and there's something that's close to the ground where it's likely colder and wetter, and something that is more dry and wind exposed, etc. So the variation within a few square meters here can far exceed the annual differences in mean temperatures on much larger geographical distances. So there's some reason to, I think, to think about how insects and other small ectotherms might navigate in these heterogeneous environments. So I'm going to present to you 3 cases, and already now I can admit that I have more questions than I have answers. But I think at least I hope that I can convince you that at least in some cases behavior is something that we need to think about. So the first case, we have considered thermal preference in Drosophila. And if we allow flies, for instance, to distribute along a thermal gradient, so we have a gradient here from around 10 degrees to just over 30, where the flies can move around freely. Then they distribute not uniformly, but according to this, to a curve that could look like something like this, where we have an overrepresentation of individuals at a certain temperature. In this case, this is what we can call the preferred temperature. So most animals assemble around this temperature, in this case, 25 degrees. And in this study, we try to start looking at preference in order to know more about how life stages and different species and different acclimation temperatures would affect this preference. So we have 4 different species were selected to be some temperate species and some more, that's for instance Drosophila subobscura, and some tropical species, Mujavensis, Drosophila melanogaster is a cosmopolitan, and Birchii is also a more warm-loving species. And we measured on 3 different traits to the left, and I will show you better pictures later, so don't worry too much about that now. Just to show that we can get distributions for the distribution of adults here to the left. In the middle is the distribution of where the same adults lay their eggs, and to the right is the distribution of larvae of the same species. And right now, I just want you to notice that these distributions are not necessarily the same. If we, for instance, look at subobscura, then we can see that the adults have a very shallow distribution and a preference around here that's between 15 and 20 degrees, and a very different distribution for where the same animals lay their eggs, which is closer to 25. We can also see that there are some problems with this, because this warm-loving Birchii species seem to have a huge preference for very low temperatures, and these are an artifact of the experimental design, because some of these animals actually wander into the cold end of the gradient and get stuck there. It simply gets so cold that they stop moving. So somehow this species do not possess the ability to assess the environment, whether it's a good idea to walk into these very cold ends of the gradient, which other species are able to. So that's also an interesting observation. I think that the species have a different ability to sense and respond behaviorally to the environment. So if we consider where these species come from, then we can calculate the annual mean temperature of the environment of their habitat. And we've done that for these 4 species that we looked at, and then we have done it for incorporated data that we have collected from the literature. So the triangles is from the study we have done, and the circles is from literature. And on the y-axis, we have plotted here to the left the adult preferred temperature, the mean preferred temperature, and to the right in the B panel, we have the juvenile, that is larvae. And in both cases, the dashed lines represent the full data set and shows a non-significant correlation between the temperature of a region, meaning that warm adapted species do not have a different preference temperature than cold adapted species. The same goes for the juveniles. If we remove the one species that had this problem of walking into a low temperature and getting stuck and thereby creating a falsely low preference, then there is a significant correlation in the solid line here between the annual mean temperature, that means the temperature of the environment these species origin from, and the preference, but it's still quite shallow. So there's no strong correlation in the preference. It's quite similar, the preference, both in juveniles and in adults, and particularly in juveniles. It's very shallow. We had, from another study for the same species, the same 4 species we investigated here, estimates of the optimal temperature where the collated developmental time, ecto-adult viability, and fertility at different temperatures was collated in order to create a performance curve, and from that we could estimate the optimal temperature. And the optimal temperature for these species is now plotted on the y-axis and the preferred temperature on the x-axis. And the closed symbols are adults themselves, and the open symbols are where those adults lay their eggs. So along the line of unity here is where there is a correspondence between the preferred temperature and the optimal temperature. And we see 2 different patterns here. The one pattern of the open symbols, that's the egg laying. It's quite condensed and closer to the line of unity. This means that all 4 species lay eggs at around the same temperature. And it's there's a good correspondence between where the developmental time is short and the survival is high in these species, and the temperature where the females want to lay their eggs. So in the eggs where the females choose to lay their eggs, they seem to prefer a temperature that matches their optimal temperature. But for the black symbols, there is both a huge variation between the species, and they all fall further away from the line of unity, here to the left, always above the line. And that means that the animals actually prefer to hang out at temperatures that are much lower than the optimal temperatures. In the same populations, in the same species, between where the adults hang out and where they go and lay the eggs. This means they actually stay at lower temperatures, then they walk up to higher temperatures, lay their eggs, and walk back. And why do they do that? We don't know for sure, of course, but the pattern is in this study that the adults prefer lower temperature than the optimal ones. And one interpretation is that this is to decrease the risk of overheating. So the thermal performance curve looks a little bit like this distribution that I showed you before. And if you are at your optimal temperature, then the fitness cost of going higher is quite dramatic. So that adults prefer lower temperatures than the optimal could be interpreted as decreasing the risk of a huge fitness cost. We also think that adults could rely more on behavior. They are able to move around more freely than juveniles. So adult flies can fly or walk quickly to other microhabitats or other microclimates, and that might mean that they can rely on behavior to control their performance. The adults, they go and lay eggs at higher temperatures, and it potentially is because that the eggs and the developing stages, they depend more on physiological mechanisms and tolerances, as their behavioral responses are more restricted. So the reason the animals walk to higher temperatures to lay eggs might be because those eggs and juveniles develop faster at higher temperatures, even though there is maybe a risk. But the tolerance might then be higher through physiological mechanisms. I didn't show the results because it didn't show much, because we did see very limited acclimation responses in preference. There is some variation among species, but regardless of whether you are acclimated to 15, 25, or 28 degrees, the preference is more or less the same. And as I said, I have less answers than I have questions, but at least I hope that you are convinced that the relationship among life stages is interesting here and could be important, and therefore needs to be studied more. Here's a related second case where I will discuss a little bit how behavior interacts with evolutionary adaptation and physiological responses. And we start out with this model where the dashed line represent the environmental temperatures available in the habitat where an animal lives, and we just made a Gaussian distribution just to have something. It does not necessarily look like this, but this is the dashed line. So if the animals hang out in this hatched red bar, then the animals will get a temperature of the body that is close to the environmental temperature. And if the solid line depicts what is the performance or the fitness curve for temperatures for this animal, then they are here just below the optimal temperature, but at a high fitness. So in this case, the animal has a good match between environmental temperature and body temperature. Then let's assume that the temperature increases. So the first thing that will happen if the temperature increases in the environment is that the dashed line will move to the right, so that we see down here. This means that the animals now suddenly are moved into an area where the available temperatures in the environment are not very good for these animals. So you can see the fitness curve here decline in the area where there's a lot of environmental temperatures available in this area. So what can happen is that the animals respond either through plasticity or evolutionary and move their tolerance upwards. In this case, the increasing body temperatures of the animals that again now match the environmental temperatures, then allow the animals to attain a good match between environmental temperatures and body temperatures. But that means that they have to move their preference so that they now are able to perform at a higher temperature. And that can only happen if the preference for temperature is quite weak. If we, on the other hand, have increasing temperatures in these dashed lines, but a strong invariable preference, so the animals, they try to stay at the same temperature as before, then there's very little available temperatures that are suitable in the habitat. But if they are there and the animals can utilize them, then they can attain the same body temperature as before here in the solid line. This in turn prevents selection for alternative tolerance phenotypes. And that means that this strong preference behavioral response or lack of behavioral response will actually slow down evolution, and it will prevent induction of adaptive plasticity. So why is this important? A lot of studies on the effect of climate and changing climates addressed through big models where these models try to predict performance on animals. And the parameters of these models are based on data that come from the lab. So for instance, whether animals are able to lay eggs or survive at a certain temperature. And the way this is typically done is that we force animals to behave at a certain temperature. So in this panel to the left, we have investigated egg-laying capacity at a forced temperatures. So we take animals, put them in vials, and we stick them at 19 degrees, and then we count how many eggs do they lay. We can do the same at 25 degrees, 28, and then 15, and then we get some kind of reaction norm or performance curve, and then we can say, yes, they lay more eggs at higher temperatures, but they still lay some at 20. And that can then be used to calculate how much the effect of increasing or decreasing temperatures can have in a certain model. But if we do the same in a voluntary assay, where we use, for instance, a gradient, where we can see whether flies will actually lay eggs at 19 degrees, even though we know from this left panel that they are physiologically able to do it, then we can see here to the right that they actually don't do it. So they definitely have a preference that even if they can lay eggs, they choose not to do it, and they choose to lay them at their preferred temperature at slightly higher temperatures. So models based on these data from capacity, physiological capacity of forced performance, they can have various consequences in estimating the effects of changing climates. So if individuals are able to exploit microclimates, then it's possible that we overestimate the negative consequences of climate change and extreme climate events, because these animals will never experience the temperatures reported because they're able to compensate by their behavior and their preference. However, even if they can, the costs in time and resources of searching out and maintaining the preferred body temperatures, they might increase as the difference between the optimal temperatures and the actual temperatures environments gets more and more rare. And therefore, we can have a strong fitness consequence that might be underestimated if we are not aware. So again, I hope I have convinced you that this behavioral response, even if quite simple here, could have a consequence for the way we understand thermal biology and the effect of climates. So the third case is from a different system, not Drosophila anymore, but from a social spider from the genus Stegodiphes. And here we have two populations collected in Namibia and Botswana. So this is the southern tip of Africa. And to the right in the B panel, we have some models of temperature data from these two localities. So in both places, it gets warm, but in Botswana, which is represented by the blue color, it is warmer, especially in the colder part of the year. So it's more uniformly warm in Botswana, and in Namibia, which is the orange, then it's considerably colder, and the more purple color here is the overlap between the two. So these animals live in nests that are exposed to sun in these bushy habitats, and nest temperatures can exceed 55 degrees inside, where most animals usually reside. And it has been observed that when it gets really warm inside these nests, then animals might move outside and sit under the nest where there is shade and maybe cooler temperatures due to wind cooling. We also know that they can't sit there all the time and do get exposed to high temperatures. So we also expect these animals to have some kind of physiological response. And based on what we have just seen, then we could imagine that behavior and physiology might have some kind of interaction that we could be interested in investigating. So let's start with the simple first, the critical thermal maximum. So the heat tolerance from the two populations. We actually measure a higher CT max in the Botswana animals, which are from the warmer environment, compared to Namibia, which is just warm. So that matches our expectations if physiology is important. and if that is the mechanism they use to endure these high temperatures. But we also tried to explore this escape behavior. So we allowed 5 individuals, juveniles, to make a nest at the closed end of a shell vial. So the vial extends upwards here towards the lamp. Then we have temperature thermocouples, and we seal off this top part of the vial with black tape. And then we warm it from the top and see when animals crawl out. And then we can correlate the escape temperature with each individual to simulate what's been observed in the field. And we have done that for a number of replicate nests from each population after acclimation at 19 and 29 degrees. So for Namibia, we find in general that both acclimation treatments escape at a higher temperature than the Botswana ones. And the Botswana ones are the warmest population. So maybe to turn the argument around, the animals from the warmer environment escape earlier at a lower temperature. And we see the same tendency in the response to acclimation, that acclimation to 29 degrees, the red boxes here, the box plots, they escape at a lower temperature than animals acclimated to 19 degrees. So at first this might sound or look counterintuitive. But the response is in the same direction, and the interpretation here is that animals that are either acclimated to high temperature or evolutionary adapted to higher temperatures, they have a behavioral response that make them escape earlier when it gets warm. And maybe because when it already starts to get warm, then it might get even warmer later. So that spiders from the warmer population has the highest CT max, that is consistent. It's not proof, of course, but it's consistent with an hypothesis of local adaptation in the genetic part of this trait. But that spiders that previously have experienced higher temperatures across within generations escape at the heat sooner is consistent both with local adaptation and plasticity in this behavioral trait. So if you only measured one of these traits, either the physiological heat tolerance, the CT max, or only the escape behavior, it might be very difficult to interpret these results. So if you are ignoring the behavior, or if you are only measuring the behavior, then you might reach incorrect or inadequate conclusions about the thermal adaptation and the role of the temperature in these animals. So I hope I have convinced you that this is important to consider behavior as well, in addition to physiology, and want to extend a huge thanks to all my co-authors and funding agencies, none mentioned and none forgotten, and thanks to the organizers for allowing me to speak.
Thank you, Jesper, for such a nice talk. It's nice to see now the behavioral side of the story. And when we have some questions already, there is one question about if you have seen a different temperature preference, if you have considered summer and winter variation for adults.
We haven't done those experiments, and for a lot of the animals that live where I live, then summer and winter is different in a way that means that we don't have active animals in winter, right? So they would be in some kind of diapause or in a overwintering stage that is different. But certainly for more southern temperate species, and of course species in even further south or north, depending on where you're at, then there could be that would be an obvious hypothesis that this would exist, and I could even imagine responses in both directions, right? That if there's some kind of acclimation response, then in the winter you would have a preference for a higher temperature. But it could also be like we saw in the spiders, that in the summer finding the cooler temperatures would be more important in order not to overheat, and therefore you could have this compensatory response. And this is not the first time it has been seen. So behavioral compensation, higher temperatures leading to preference for lower, has been seen in other places as well. So absolutely, that could be something in that regard.
Yeah, there's another question. Thank you for the answer about if this pattern of different like physiological response, and when you present a choice for the adults regarding egg laying, for example, have you seen something alike for the larva stage? Because they can move also. I mean,
yes. So we did do larvae, and what it's a little bit more tricky because they are slower and then they are developing fast, and that is tricky. But it seems as the larvae, at least for the data from this study I presented, the distribution is more shallow. So it's not as clear a peak as it is, for instance, for the egg laying. So the sharpest peak in preference seemed to be the egg laying. And this was also the case where the 4 different species had the most similar preference. So all the species laid eggs around 25 degrees. So there seemed to be some kind of, or at least based on this limited number of species, maybe a basic physiological optimal temperature for this life stage, at least in this genus, or at least across those 4 species. I think that for the larvae, I don't think it's so prominent for this species, but from other species there is a lot of other types of behavioral patterns that might be more important. And also for Drosophila, to some extent, we know that females might respond to the egg laying of other females, right? So if one female starts to lay eggs, then there will be some kind of hormonal cue that might attract other animals to lay eggs there. Maybe it's a good place, or maybe it's a place to avoid, right? And we also know that some species aggregate the juveniles and maybe increase the temperatures around where they are, and they might actually prefer better quality food over optimal temperature, etc. Right? So if there's a low quality food in the middle of the gradient and higher quality food at the ends, then flies will actually go away from the preference to lay eggs where the food is better. So there's a lot of layers here that where these complex decisions of these animals, it's certainly not random where they are and how they lay their eggs, for instance.
Yes, certainly decision making is important here.
Yes.
Well, there's another question here from Mats. He says, hi, is the thermal performance curve from the voluntarily egg laying then more appropriate for modeling or future species distribution responses, for example, under climate warming? Doesn't it underestimate their capacity?
Yeah, I don't know that it's more appropriate or less appropriate. I think it's a question that we need to think about. What is appropriate? Because it's of course also so that if temperatures increase and there are no 19-degree available at all where there's adequate resources, then flies will go somewhere else and lay their eggs, right? So I think both the preference and the capacity are important aspects to consider. And I don't think that we know enough, and that's also why the title indicated, and I indicated earlier in the talk that I have more questions than answers. I think at least that we can point to some papers, also influential papers, that have done models on data that are naive and that make predictions that are not at least fully enlightening for everything we know.
Yes, as you said, there are more questions than answers.
Absolutely, yes.
Well,
but that's good. We have young scientists that can start working on these questions.
Yeah, for sure. And that's the purpose of this event, that it could inspire this early researcher. Well, thank you, Jesper, again for the nice talk. And now we will have a short break of 5 minutes, and then we will continue with the next and last speaker of the day. So thank you very much again.
You're welcome. Thank you.
Well, it has been a very great event indeed, and now we are going to watch another talk, changing a bit the scale of body sizes here, and I am very pleased to welcome Dr. Matt Schou. He holds a PhD from the Department of Biological Sciences at Aarhus University, but he currently works as a postdoctoral fellow in the Department of Biology at the Lund University in Sweden. His research is focused on how diversity among individuals in a species evolve, and he's interested also in aspects of cooperation and stress in South African ostriches, which is what he's going to talk about today, and evolution of environmental sensitivity. So Mats, thank you very much for your presence here today. After your talk, we will have again up to 10 minutes to take questions from the audience on YouTube. So thank you very much. Mats, we cannot hear you or see you.
Can you hear me now?
Yeah, that's okay.
Good, I'll try again. Thank you for the nice introduction, Gustavo, and I'm very happy to be here. As you said, I'm not working on small ectotherms, but I try to pick a picture of my study organism where it looks very, very small. I don't know if I succeeded, but I try to. But I still think there's a link to the small ectotherms, because I think that all the excellent work that has been and are being carried out on small ectotherms, such as Drosophila, is really providing the basis needed for the studies that I'm doing right now at ostriches. And I did myself PhD on Drosophila, and I see myself now as actually using all that know-how and the frameworks and the methods to study thermal adaptation in ostriches. Because the ostrich is a vertebrate, and compared to small ectotherms, I argue we know very little about the thermal adaptation of vertebrates. So yes, I'll start with a bit of introduction in terms of my motivation. So for most species groups, we can find species that are adapted to the most cold temperatures, and species that are adapted to the hot, maybe the deserts, and species that are present in more temperate regions. And this goes also for the birds, which I've used here to illustrate this diversity that's created amongst others by adapting to different temperatures. And as I said before, most of the work that has been carried out on vertebrates, such as the birds, has focused on the physiological, maybe the morphology that has enabled organisms to adapt to heat or to cold stress. So focused either on heat, for example, focusing on the heat adaptation of birds in the desert, or focusing on the cold adaptation of penguins in Antarctica. There's also been loads of work done on the temperate species of birds, but their focus has been on the timing of reproduction, not the tolerance of it in terms of varying temperatures. But as I think both Volker and Jesper touched upon in the previous talks, these animals are not only exposed to the mean temperature in their environment, but also to large fluctuations in temperature. So they all have to cope with not only the mean, maybe hot temperature for the deserts, but also the low, very, very cold nights and just the cold days that does happen. They need to cope with these as well. And this fluctuations in temperature has been, known for very, very long time, right? Here's illustrated by a classical paper by Sørensen with 2 mountain passes and the fluctuations in temperature that are both across the year, but also say within days or weeks, within different months. And if we highlight maybe the reproductive period of most species is right bang on in a period where there actually is loads of thermal fluctuations. So these animals, they have to cope with temperature fluctuations also in the reproductive period, which is maybe the most crucial period for their persistence. And therefore, that's what I'm studying. So this leads to the first and maybe the overreaching question of my work on ostriches: how is reproduction affected by thermal fluctuations? I'll answer this, and then I will come up with 3 follow-up, or I will try to answer 3 follow-up questions that I have, leaving a bit of suspense, trying to anyway. Here, quick picture of a beautiful little lady ostrich sitting in a desert in South Africa. And the reason why, or one of the many reasons why I'm studying ostriches is that its distribution overlaps with some of the regions in the world which has high range of daily temperatures. So it's really exposed to a fluctuating environment. Another thing that characterizes the ostrich and other tropical and subtropical birds, is that it actually lays many eggs across the year. It's not just one clutch as we see in temperate species, which makes it difficult to really study small-scale effects of temperature on reproduction. The ostrich is a ground nester, maybe not so surprising given its size and lack of flight. And it likes to lay eggs in a nest in the sun, not in the shadow, probably because of predation risk, but that's anecdotal. And it can lay, so one single female can lay multiple nests, and nests also have a really, really high failure probability because of predation. And this means that they need to lay eggs continuously throughout the year. A nest might fail, they have to create another one. They might come across a nest of another female that they can lay in. So they lay all the time, which is a great opportunity for us to study small-scale temperature effects on their egg-laying rate. Throughout the reproduction, there are different phases of reproduction, and the ostrich can, as other animals, invoke different thermal adaptations to cope and through these reproductive phases. And it might be different mechanisms that are important for different phases, like the panting might be important during incubation, where the ostrich cannot get up because it would expose the eggs to overheating. But today, I will only focus on the phase of egg laying rate and how that's impacted by temperature fluctuations. And the data that this is based on is from a research farm in South Africa we are collaborating with, where they have been monitoring egg laying rates of ostrich pairs for the last 21 years in almost 200 enclosures, which you can see at the satellite image in the middle. And there's one male and one female in each enclosure for most of the year, and whenever they lay an egg, it's recorded. And that gives a total of around 600 females, which we can connect with the pedigree. And each female has been monitored for several years. So it's repeated measures of egg laying within years and repeated measures of these yearly temperature-dependent egg laying rates also across years. So it's a strong data set to investigate the temperature effects on reproduction. So let's get back to the question: how is reproduction affected by thermal fluctuations? Before just correlating egg laying rate with temperature, we needed to figure out, well, what time period, what is the critical thermal window that affects egg laying the most? And we used the sliding window approach to figure this out. And we saw that the probability of laying an egg or not laying an egg at a given day is determined by the average temperature 2 to 4 days back in time. And this actually makes, at first it seemed a bit surprising to us, but it makes quite a lot of sense, because it takes approximately 2 days for the ostrich to produce the egg. So after making the decision to, okay, this is a good habitat, good temperature, I want to make an egg, it takes 2 days before it can actually lay the egg. So with this information, we could then investigate the relation of the impact of this temperature on the egg laying rate of ostriches. And this is depicted here in this graph to the right. and as you see, there's a strong impact on temperature on egg laying rate. There's an optimum egg laying rate around 20 degrees. And if temperatures are decreased from this temperature optimum, or if they increase, there's reductions in egg laying rate. So this species, the ostrich, which is adapted to living in the Kalahari Desert, and also the temperate regions of South Africa, like the Cape region, they're actually quite sensitive to temperature fluctuations. So next question from this cool find, I think, and really interesting that they are so sensitive. But for me, the interesting part is evolution, and so I immediately thought whether can this plasticity, so these slopes, this change in egg laying rate, increasing or decreasing temperature, can this evolve? So in a situation where the population of ostriches migrated to a colder an area, could they then adapt to this area and increase their egg laying rate at lower temperatures? Or in a world that becomes warmer, can they increase the egg laying rate at high temperatures? And because we have a pedigree, it's actually possible to test this. So this is the second question. Is there individual variation in this thermal plasticity? The way we approached this was to take the data that I shown before and then split it in two at the optimum and say, okay, on everything below the optimum is cold stress, everything above is heat stress. And then we can collapse this data into one single axis called thermal stress, such that any increase in thermal stress is either cold stress if it's coming from reduction in temperature from optimum, or it's heat stress if it comes from a increase in temperature from the thermal optimum. So it's exactly the same data, it's just on a different scale. But this makes it much easier to work with, and we can now, for every female, we can estimate a slope for illustrating the rate of change in egg laying rate with increasing temperatures at the top here to the left, or decreasing temperatures at the bottom to the left. So every line on the graph to the left is one female, and this is just data from 1 year to illustrate the large variation there is in how females are responding in terms of egg laying rate to change to increasing temperatures, or in the bottom to decreasing temperatures. And we can then put this data into a big statistical model, generalized linear mixed model, which I will not go more into, and estimate the individual variation in these slopes to test, okay, is there significant individual variation in these slopes, so it is in this plasticity. And we do find significant variation both for the hot slopes and the cold slopes. So some females are better at maintaining egg laying rates when it gets colder or when it gets hotter. And we can then use the pedigree to see, okay, but is there part of this individual variation which is due to relatedness amongst these females and therefore potentially heritable? And we find significant genetic variation in these slopes as well, and we can, by then dividing this by all the individual variation and also some within individual variation that we estimated as well, we can get an estimate of the heritability of this thermal plasticity, and conclude that this is a heritable trait. It can evolve, so it is actually possible. It appears, according to this data, in this population of ostriches, to evolve a greater heat tolerance in terms of egg laying rate, or cold tolerance in terms of egg laying rate when it gets colder. So some females are carrying genes that are making them better at coping with increased temperatures or decreasing temperatures in terms of their egg laying rate. Moving on to next question, that is, will it evolve? Now we've shown that it can. We have the necessary genetic variation, but for it to actually evolve and be important for the ostriches, it needs to be under selection. That's the next question. Is this thermal plasticity under selection? And we used a Same framework again. We have these slopes of the individual females. How are they responding to increasing heat stress or increasing cold stress? Then we can correlate these individual slopes with individual fitness to try to understand what kind of slopes, what kind of response to increasing or decreasing temperatures is most optimal. And the way we measure individual fitness here is using a yearly egg laying rate. So disregarding temperature, but just how many eggs does this female manage to lay across the full year. So we correlate these in a statistical model, and we could then estimate the phenotypic and also genetic correlations between cold stress slope, so the cold tolerance, and the fitness in terms of the yearly egg laying rate. And here on this graph we have, I've not shown the data yet, I will in a second, but to the right of the cold, on the x-axis we have values indicating no decline or a small increase in egg laying, and to the left we have steep declines in egg laying, so a low cold tolerance. This is the data, so the model and the data supports that there's stabilizing selection for cold tolerance in such a way that it's most optimal to have a cold stress slope of 0. So basically no decline and also no increase in egg laying rates when it gets colder. That's the most optimal for this population at the current environment. And we did the same for heat stress, so correlating these individual heat slopes with the yearly egg laying rates, and we find a similar pattern. Also, the most optimal thermal tolerance is one where you are able to, as an ostrich, to maintain your egg laying rates, not increasing them, not suffering a decrease, but maintain them. That will give you the highest fitness. So again, stabilizing selection. So with these two findings, first, thermoplasticity under heat and cold stress is heritable, and secondly, resilience to heat and cold stress is under stabilizing selection. It kind of wraps up the story, and we now have a good feeling for what's going on with these ostriches. So we can imagine that if it gets warmer, or if they would migrate to a different place, then the stabilizing selection curve would shift, and they would change their cold and heat tolerance accordingly to optimize their fitness. But as I said in the beginning, the focus here is on fluctuating temperatures and on cold and heat stress. So the last thing I wanted to do with this data was to integrate heat and cold stress, tolerance to heat and cold. So basically asking if they are genetically integrated. So can you, as an ostrich individual, increase your heat tolerance whilst maintaining your cold tolerance, or maybe even also increasing your cold tolerance? And we continued in the same framework. So maybe a bit boring, but I will repeat it again, using these slopes of change in egg laying rate of individual females when it gets colder or hotter, but then this time correlating them with each other. So correlating, taking one female and saying, is her, how is the relationship between her heat tolerance and her cold tolerance, so the hot slopes and the cold slopes, and then doing this across all the females to see if there's a relationship. And then this is what we find. This is the somewhat raw data. So this is not a model, and what we see is a negative relationship. So individuals that have a high heat tolerance, or they have a low cold tolerance, and individuals with low heat tolerance, they have a high cold tolerance. So it appears that there is a trade-off between these two traits, or they are somehow linked. And when we then run the model to estimate the correlation, phenotypic correlations and genetic correlations, we see that this is significant. So it's a negative genetic correlation, which I think means that it's constraining the ability of the ostrich to adapt in both these traits at the same time. So if it gets warmer, then it can increase over evolutionary time. their heat tolerance, but it would have a cost in their cold tolerance, which is problematic if or is challenging if we have an increase in fluctuations over time, which means that it gets warmer at the extreme. So the hot days becomes hotter, but the cold days, they don't get less cold. They stay the same, right? Then it will be challenging for these ostriches to shift evolutionary to adapt to this. Before finishing off, I will just present data from a few other populations, because we wanted to make sure that this finding was not just specific to this population that we have been following at this site, and we actually at the site in South Africa we also have individuals from two other populations called Zimbabwean blues and the Kenyan reds. And I'm sure you can all see why it's called Kenyan reds. It's not angry; it's just red. That's how it looks. And for these populations, we didn't have so many individuals, and that's why they were not included in the pedigree-based analysis, because we don't have enough individuals to look at the genetic correlations. But we could estimate phenotypic correlations between heat and cold tolerance for these populations as well. But before doing that, I'll just quickly show their egg-laying rates compared to the population that I've talked about until now, which is the gray dots on this graph, because I think these population level lines here also illustrate this potential trade-off between heat and cold tolerance, because you can see that the reds, which are illustrated by this orange color, they have maybe a more steep decline in egg-laying rates when it gets cold, but on the other hand, they have a less steep decline when it gets warm compared to the South African blacks, so the gray dots, right? So at the population level, we also see this trade-off, which might also actually illustrate that these Kenyan reds have adapted to a warm environment up in Kenya by increasing their resilience to heat, and that has cost them their resilience to cold, but they have to find the right balance for their environment. And when we then estimate or correlate cold and heat tolerance for these populations, we see similar negative correlations as for our original study population. So this is a general phenomenon across populations, not just specific to ours. I should say here that the reason why the error is so little on this plot is because it's output from a model. So it's predicted data points, it's not the raw data. So I think I made it on time. A quick summary. Egg-laying rate is reduced at low and high temperatures in the ostrich, but there's individual variation in the extent of these reductions, and this is heritable. So it means it can evolve. And there is a selection forward stabilizing selection for maintaining your egg-laying rates as much as possible across different temperatures. But the plastic responses to heat and cold are genetically integrated via tight negative linkage. So I'll just quickly say thank you for having me, and thank you to my collaborators and some funding agencies.
Well, thanks to you, Matt. It was quite a very nice presentation of yours. Now we will take the questions from the audience. There is one question here saying, considering the long lifespan expected for an ostrich, is there any known or expected effect of aging in the interaction between ambient temperature and egg-laying behavior?
Yes, they can live long, and I don't know. I mean, I can say, I mean, there hasn't been done any studies on it, but there have been studies on how egg laying is affected by age, and they start laying eggs when they're around 2 years old, but they lay much fewer eggs when they're 2 than when they get to 3, and then it's stable as they age. Of course, with some upper limit, but we have very little data on that. So in these analyses, individuals that have age 2 have been removed because there could be such interactions, but I don't think we have the power do that, but we haven't tried. So we could actually do it. It could be interesting if the young ones had a different laying strategy, maybe. Yeah. Was that? I hope that was an answer. Yeah.
Yeah. Well, yeah. I actually was wondering something on my own about that, because you showed that there is a considerable among individual plasticity on egg laying rate, and I was thinking if you know of any other physiological or morphological correlates that could explain, or at least be associated to these differences on egg laying rate.
Yeah. There are. I mean, so we did some initial studies where we looked at multiple traits of reproduction. So also sperm in males and egg mass, hatching success, and so on. But we found very little, if I remember correctly, correlations between these traits. At least not very strong. So that's why we stuck with egg laying rates for further analysis.
Okay. There's another question here. I think that there's considering climate change effects, how would you consider that ostrich egg laying rates would be affected in warmer environments? I mean, regarding climate change scenarios.
So if it just gets warmer, right? If the entire thermal range just increases, so it gets less cold and more warm, well, then I think the data shows that they are like more than capable of adapting and increasing their cold tolerance, or reproductive, you could call it, cold tolerance. It's more a challenge for them if the hot days become even hotter, whilst the cold days they just stay as cold as they've always been, right? Which is some of the predictions that have been made are like this: increased mean temperatures, but also increased fluctuations. Then they will not be able to track climate change in terms of egg laying rate. And that's at least what my data suggests. But I will also say, in general, the ostrich is capable of laying like at extreme temperatures, right? So I'm sure the species will be fine. Here, in this context for the ostrich, I think it's more about maybe has to move out of some parts of the Kalahari and migrate a bit around to different habitats. The importance of this finding for me is more if this is general across other species, then it gets interesting. But I don't know, of course.
Yeah. Well, you show also some like intraspecific differences when you compare different populations, but would you consider that, I mean, would you be expecting that these were greater, the differences among populations? Because it seems that the response is quite conserved between the populations you studied.
Sorry, I didn't get the question.
Yeah,
can you repeat?
Yeah. When you compare the two different populations, the difference seemed quite low, if I am not mistaken.
The differences in the egg laying rates, or do you mean this correlation between heat and cold?
Yeah, that response is quite general.
Yeah,
so
yeah.
Is it because this is a constraint within the response to cold and heat? Is this a general response within the species? Is that it?
I think it's, my best guess is that it's some genetic constraint. You could maybe think of morphological traits that would increase your ability to endure heat, like maintain a low body temperature, which would then cost you when it gets cold. For example, increased insulation. Like if you're more insulated, then you might suffer when it's hot for a very long time, but you might benefit when it gets really, really cold. And I think there are multiple traits like this that are not, so traits that are not very plastic, if they are genetically encoded, and that's what we are seeing variation in, then that could explain this tight negative linkage. Or it could be molecular cellular responses to increased body temperatures. It could be genes related to production of eggs, so turn on, turn off of egg production. It could be many things, but yeah, for me, it's a constraint. But what type is very difficult to say.
Yeah. Yeah, sure. There's another question, and I think it's regarding now the difference between population in egg laying rate. If it is, do you think it's the average or daily minimum maximum temperatures across the year that influence these differences observed between populations? And yeah.
I don't know. I mean, there are many differences between the habitats. I think up in Kenya, as I remember it, it's warmer up there, but there's also, I think, a greater seasonality in temperatures, less precipitation. I think there's many things, and we have two populations. I think the previous speakers knows that you need 25 species at least if you want to say something about that. So I will refrain from saying anything, but of course it's nice to speculate. And I think we do, this is soon published, and I think we do speculate a bit in the discussion just because you have to.
Yeah, sure. And I was thinking, do you know if there is any correlation between body size of the ostriches and the egg laying rate? I mean, are differences among egg laying rate explained by differences in body sizes among females?
Not that I'm aware of, no.
Okay, and all the individuals in the big population, these 60 and something hundred of females, are similar in body size.
Not similar, but I mean, what's similar, right? There's of course variation. But the body mass also changes over time. So they actually during the season where they lay eggs, they lose weight, and then in the couple of months, or 4 months, where they are not kept in pairs but kept as a group of individuals, then they're regaining weight. So it actually fluctuates quite a lot.
Yeah. Okay, then I think there's no more questions here. So Matt, once again, thank you very much for the nice presentation and for your answers. And I take this opportunity to thank very much all speakers for these amazing talks, and all of you that are watching through on YouTube. We invite you to join us tomorrow for the second and final day of this event and of the Physio webinar spin-off this year. And so please follow us also on our social media, and yeah, see you all tomorrow.